My presentation is just going to, I guess, demonstrate how you can deconstruct the RSD to make it not so much an assessment rubric, but a design rubric that we've actually been using to design our flipped classrooms. Everyone familiar with the flipped classroom concept?
No.
Anyone not familiar?
I'm not familiar.
Okay, the flipped classroom concept is a way in which the homework elements of a course and the face-to-face are actually reversed. So you give students some, work to do at home so they can prepare for the actual face-to-face that becomes a lot more interactive. So hopefully, by the end of this session, you'll see how we've used the RSD to design our flipped learning activities in a first-year human biology course, and then how it's actually helped us to harmonise the pre-face-to-face with the face-to-face segments of learning. So what we actually did was map the different sequences, and if you have a look here, this is our modified version. When we look at the extent of students' autonomy, we're looking at the pre-face-to-face, which is the level 1 in the research component. That is basically where we want students to do their pre-classroom material to get a grasp of the threshold concepts. By the time they come to the face-to-face, they're working in level 2, and then when you start to give them their sort of set assignments, they move into level 3. So as an example of level 1 prescribed research, we usually set a pre-recorded interactive module that's really short, and we chunk it into about 10-minute slots, and students either watch videos and then have to answer some interactive questions online. But we give them a lot of instructions, and we prepare them, and we induct them into this way of learning. When they actually answer their questions online, we make sure that we're using a format, whether it's through your learning management system or whether you use SurveyMonkey, that actually gives us information on their level of understanding, based on their responses to the questions that we actually set. So this pre-recorded and pre-classroom activity has to be fairly simple and an opportunity for the students to be able to work out whether or not they understand the basic concepts. So when they come to class, which we then take them to the level 2 of the rubric, we get them to do a lot of group work where they can then clarify a lot of the information as well. So in this instance, we get them to draw, make posters, conceptual posters on what they see as each, in this instance, it's, the structures of a cell. So they need to sort of look at each little structure and how it's represented or how they perceive it. After that, again in the face-to-face, we get them to do a little matching exercise where they have to start applying that information at a little bit of a higher level. So we give them some histological photos of cells, and this is, in the first few weeks of studying human biology, and some of them have never done it before. And then we give them a little bit about the function of the cell, and they have to almost match up the form to function, which takes a little bit of application. Once they've actually got their head around the threshold concepts, we move them then into level 3, which is your post-face-to-face learning. And this is where we set their summative assessment tasks. So for instance, the first one is a written report that they do in semester 1 on the biological effects of radiation as it pertains to cellular activity. So they've looked at the basic concepts, they've applied that information, and now they're sort of taking it a little bit further. Then in semester 2, we get them to do another group wiki project. Again, around the level 3, we don't really take our first year students beyond that. So by this stage, they can now start to frame their own questions and go out and organise their own information, synthesise their own understandings, and then apply those understandings to a real-life concept in oral health practice. And we get them to produce a poster that they then have to present to their peers. In second year, we still use that same type of flipped scaffolding of the RSD, and we take them, say, up to level 4 when they look at presenting a written clinical case study. So, the RSD, I guess, has helped us design the learning, as well as using it as the assessment rubric to assess the different stages of their learning that we've actually designed. And then when they get to third year, which takes them right up to, the open research area, they then find their own area of interest, and it's basically where they do their capstone project and have to initiate their own research, their own questions, and almost create those new understandings that will contribute to their discipline in the future. So that's how we use the rubric to create our flipped classes. Sorry, thank you.